And next, we're going to review the questions from the DNA, the biotechnology reading. Number 1 says, in order for cloning to occur, what has to be removed from the donor egg? Well, they need the genetic information from the donor to implant into the surrogate mother. So that would be contained in the nucleus. So they remove the nucleus from a body cell. They can't use an egg cell from the donor because it would only have half the genetic material. They have to use a body cell. So they take the nucleus from the body cell of the donor and implant it into the surrogate mother. So that would be nucleus. The nucleus is contained in the DNA. What are the small circular pieces of DNA in a bacteria called? This little circled pieces of DNA are called plasmid, and that's what they're going to splice open to make the recombinant DNA. It's the plasmid. Oops, spell S-M-I-D. Number 3. What is the name given to the identification of all genes found in the human DNA? Identification of all the genes is called the Human Genome Project. It's called the Human Genome Project, and it was an effort to identify all of the genes in the human genome. It's a Human Genome Project. And it was just recently completed. Number 4. Which of the following statements are true about genetic engineering? Pieces of DNA from 2 different organisms are joined. Yes, if we're doing recombinant DNA. Genes from complex organisms like an animal can be injected into a yeast or bacteria. Yes. Recombinant DNA is passed down to the offspring during reproduction. Yes. And number 4, that is, it is used to produce large amounts of proteins to treat certain diseases. And that is true. So all of those are true. Number 5. What process can be used to separate DNA fragments of different sizes? That's how they identify the little fragments between the actual genes. It's called the DNA fingerprint. It's unique to an individual. No 2 people, except for possibly identical twins, have identical DNA fingerprints. That's called DNA fingerprinting. Also called gel electrophoresis. They cut the DNA into little fragments, different little pieces, and they can put it into a gel tray. There's little wells that you can inject the particles of DNA into, and because DNA has an electrical charge on it, they can apply a charge to the tray, and then the particles will move through the tray, and move through the gel in the tray. Because the particles are different sizes, they can move different distances. It's harder for the longer pieces to move through the gel. They have a lot more resistance, so they can't move as quickly as the smaller particles. So the smaller particles are going to move further down, and it leaves a banding pattern. Number 6 says DNA molecules that are made by inserting a sequence from one organism into the DNA of another organism. So you're going to take a piece of DNA and put it into another piece of DNA, and that's going to make what's called recombinant DNA. You're going to recombine those 2 DNA particles together. It's called recombinant DNA. Recombinant DNA. You recombine those two pieces of DNA from two different organisms. Recombinant DNA. Genetic engineering is a method used to, the main reason you do genetic engineering is to produce organisms with desirable traits. You're going to want to remember that. Genetic engineering is a method used to produce organisms with desirable traits, whether you're wanting to make teeny tiny little dogs out of big dogs, or whether you're wanting to make seedless watermelons, or whether you're wanting to make a tree that grows really big red apples. You're searching for a desired trait, and you're going to breed two offspring with that trait that you want. Take their offspring that's got that trait, breed them again until you get it where the majority of the offspring will be having that trait. Okay. You're producing offspring with desirable traits. Whole reason for doing genetic engineering. In cloning, the offspring is genetically blank to the animal that donated the nucleus. They're going to be genetically identical. Just taking the entire DNA material from one cell and placing it in another, so it's not being modified. It's going to be identical to that original DNA sample. Through gel electrophoresis, DNA fragments are separated according to their size. That's what I was saying just a minute ago. It's hard for the bigger pieces to squeeze through that gel, so they're not going to go as far. The little pieces will go farther, and then that leaves a banding pattern. They're separated according to size. Number 10, the pattern that results from gel electrophoresis is known as, that's the DNA fingerprint. It's known as a DNA fingerprint. DNA fingerprint. Matching. Protein essential for monitoring the level of sugar in the blood. Insulin is a protein that your body produces to control your blood glucose levels. So that would be C, insulin. So that would be C, insulin. Genes or pieces of DNA are taken from one organism and transferred to another organism. So genes or pieces of DNA taken from one organism and transferred to another organism. That would be genetic engineering. Number 13. The use of organisms or their products to improve human health and food production. That would be biotechnology. Biotechnology. Number 15, genetically identical individuals are created from a single cell, and that process is called cloning. Number 15 lists 2 advantages of genetically engineering human insulin. Again, that's back to our bell ringer. Genetically engineering bacteria to produce human insulin increases the supply of insulin and lowers the cost. And you also get some other benefits such as insulin for a long time has been grown in pigs, so if someone was allergic to pork, they would not be able to use pig insulin. And there's also, it lessens the chance that there's contaminations and things in the insulin. 16 says, list one advantage and one disadvantage of the Human Genome Project. Well, one advantage would be it can be used to diagnose genetic disorders. Which could also be the disadvantage, because if you know that maybe you have a gene for something like Huntington's disease, which is a rather devastating illness, but you're perfectly healthy now, you might not want to know that 30 years from now that you're going to have Huntington's disease. Or you would not want your insurance company getting a hold of the information that you might become ill later on. So the advantage could also be the disadvantage. It can be used to diagnose genetic disorders, which might mean you can get treatment for those disorders earlier. But also, there's some ethical issues with knowing ahead of time. So some ethical issues with being able to see into someone's genome. This one advantage of cloning. Well, one useful thing about cloning is that it can increase the number of endangered species. It can increase the populations of endangered species. Don't know what's happening here. Let's see. So the advantage of cloning is it can increase the numbers, or the population can increase the population of an endangered species. Number 18 says, why do law enforcement technicians use DNA fingerprints to help identify someone who may have been at the scene of a crime? Two advantages of genetically engineered crops such as wheat is improved nutrition and improved crop yield.